Hello everyone. This week in lab we're going to talk about bacterial smears and we're also going to talk about simple stains. Later we're going to talk about different type of stains, but in this PowerPoint we're going to concentrate on talking about simple stains. So the reason why we stain bacteria in the first place is because bacteria cells don't have a color. So for us to be able to view them under the microscope, we will need to stain them so that we would be able to see them. Now, the stains that we use, or you can refer to them as the dyes, they basically add color to the bacteria cell, as I said, so that they can make them visible under the microscope. So the first step is you would want to transfer some bacteria onto a glass slide. So you would have a slide right here. Now, we usually add just one sample of bacteria, but sometimes you can actually add several samples all on the same slide. It's not recommended to do that, but sometimes if you want to save or you don't want to have so many slides and you want to save up on your resources, you can go ahead and kind of use one slide and put multiple, multiple different samples on it. Now, if you're going to put multiple different samples on it, you can go ahead and use like a crayon so that you can go ahead and draw circles on your slide so that you know that, okay, within this circle, I'm going to put sample A, in this one, I'm going to put sample B, in that one, I'm going to put sample C. Now, if you're not going to put multiple samples, we usually do not draw these circles, but sometimes, especially for students who are let's say it's their first time making a slide, sometimes drawing that circle does help them, when it's time to find the specimen under the microscope. So they know that they need to like focus right here within that circle that they've drawn. But as I said, if you're only going to put like one sample of bacteria on your slide, you don't have to draw the circles at all. So after you go ahead and you transfer your bacteria onto your circle, we're actually going to talk about how do you go ahead and prepare a smear. But you can kind of see right here, here's a smear of bacteria right there. We're going to talk about how that smear needs to be thin. It needs to be a pretty thin smear, and it needs to be evened out. Like you don't want to have your smear like over here like really chunky and then over here thin. You want to make sure that you spread your bacteria evenly onto the slides. And this is super important because if your smear is not thin and even, then you're not gonna get good results when it's time for you to view your specimen under the microscope. We're also going to talk about how to fix a bacteria onto a glass slide. We're going to talk about how we do that by passing your slide a couple of times over a flame. So we're going to talk about that in more detail in just a few. And when you come and prepare your smear, by the way, you don't want your smear to be, as I said, like too thick. Meaning that when you come and take a look at it, if your smear is super white, that would tell you that you've actually transferred a whole lot of bacteria. So compare this smear right here to this one right there in the middle. Notice how the one in the middle, it's not very white, and that's actually better. So you don't want, as I said, your smear to look something like this, very white, because that would mean that you've transferred a lot of bacteria, and that's not good because when you take a look at it under the microscope, all the bacteria cells are actually going to be stacked on top of each other to the point that you're not going to even be able to see their shape or their morphology. So the thinner the smear, the better. And again, one way for you to figure out whether it's thin or not, look at your smear. You don't want it to be very white. Okay. And then, as I said, you want to make sure that you spread the bacteria, throughout so that you don't have it, chunky. So a lot of bacteria over here, and then like you have nothing over on this side. So you want to make sure that you spread it evenly. So to prepare a thin and even smear, first thing is you want to grab your slide, of course, and you want to make sure that you clean it. So how do we clean a slide? We usually wash it with water and soap. So we wash it with water and soap, and the reason why we do that, it's because you don't want to have any dirt or fingerprints on your screen or oil. Sorry, not on your screen, on your slide, okay? So you don't want your slide to have fingerprints or dirt on it. So we usually wash it with water and soap, and then of course after you wash it with water and soap, you gotta dry it. Make sure that when you dry it, you don't dry it with something that's gonna leave, lint on it. So we usually dry it, and you can use like lens paper, okay, to dry it to make sure again that there's no lint, left on the slide. And then after it's cleaned up and everything and it's dried, we go ahead and we label the slides. So we usually put our initials on our slides. We usually put the date. So the date is very important. And also, of course, your initials. And then, you would put what type of stain are you performing. So is this a simple stain? Is it a negative stain? Is it an acid-fast stain, a gram stain, endospore stain? So basically, like put down what type of stain are you doing? And then if you know the name of the microorganism that you are looking at, go ahead and write it down. So if you're looking at E. coli, go ahead and write E. coli. Go ahead, write S. aureus. If it's an unknown, then you don't have, then you don't know what type of microorganism you might be facing if it's an unknown. Maybe if your unknown has a number. So like if if you're in the lab and we give you a test tube and we tell you, okay, this is unknown number 13, go ahead, perform some tests to figure out what type of bacteria you have. Well, okay, then you would put your unknown number, okay? But if you know what type of microorganism you're looking at, then go ahead and write the name of that microorganism. So after you, as I said, you cleaned your slides, you wrote down, and plus you wrote down the stuff on your slide. What I wanted to add is make sure that you don't take too much space, okay? So when you write on your slides, you want to kind of, write very small so that you don't take too much space on your slide, okay? Then we have to aseptically transfer our bacteria onto our slide. So of course, if we're gonna talk from the very beginning, when you come into the lab, of course, the first thing you gotta do is wash your hands, make sure that you tie your hair back, you go ahead and you put on your lab coat, put on your gloves, make sure that you clean your countertop, right? So make sure that you're doing all of those aseptic techniques before you even begin, right? And then go ahead and turn on your Bunsen burner so that you can have that sterile environment. Make sure that you are working really close to that Bunsen burner. And then make sure that you also sterilize the tools that you are about to use. So for example, right here, this is an inoculating loop. So of course, make sure that you sterilize that inoculating loop by passing it into the flame until it turns red, right? Until this whole entire part turns red. And then wait a few seconds before you go ahead and you pick up your bacteria. So usually, what we do to pick up the bacteria, now it depends where your bacteria is, where it's been cultured. So we can culture bacteria in a broth, or we can culture bacteria on a slant or on a plate. Now, if you go ahead and have your bacteria cultured in a broth, remember how we said broth is a liquid medium? So that bacteria is it's basically growing, right, in a liquid medium. So for you to go ahead and pick up some of that bacteria that's been cultured in broth, what you want to do is, of course, after you've sterilized your inoculating gloop, and that part, the whole part, it has turned red, as I said, just wait like a second so that it's not too hot. And then you're going to go ahead and grab your broth. Make sure that when you open your broth tube, you're opening it next to the Bunsen burner. Make sure that you flame the mouth, right, of the broth tube. Never lay down the cover of your broth tube onto your countertop. After you've made sure that you flamed the mouth of your broth, take your sterilized loop, stick it into the broth medium. Make sure though that you have flickered the broth first, because sometimes the bacteria settles down to the bottom of your broth. So make sure that you've shaked your broth tube, or you kind of like flickered it, okay? So that when you go ahead and dip your inoculating gloop into the broth, you are gonna, you've made sure that you are going to pick up some bacteria, okay? And then you take that loop full of bacteria, and then you put it directly onto your slide, and then using your inoculating gloop, you're going to go ahead and smear that bacteria onto your slide. Okay, you want to make sure that you are smearing it evenly. Okay, of course, don't smear the part that has your initials on it. So if this is the part right here on the edge where I have my initials, well, of course, I'm not going to smear any bacteria onto my initials. I'm just going to smear the bacteria, on the other part of my slide. And this is how you transfer bacteria that's been cultured in a broth onto your slide. Now, if you go ahead and transfer bacteria from a slant or from a plate, now bacteria growing on a slant and a plate, that's a dry medium, right? So slant and plates, they have agar, so they're kind of considered, solidified medium. So what we usually do is the technique is a bit different. Okay, so you grab a clean slide, make sure you've sterilized your loop, as I said. Then what we do is we go ahead and place a drop of water onto our slide. Okay, so you put a drop of water onto your slide, and then after you've placed the drop of water onto your slide, well, before I tell you the next step, that water though shouldn't be too much. Okay, so you don't want to add a lot of water on your slide. A very, very super tiny drop of water onto your slide, and I'll explain why you don't want to put too much water. After you put that drop of water, then you go ahead and grab your sterilized loop, pick some bacteria that's growing onto your plate and slant, and go ahead and mix it in with the water. Okay, so you're going to mix it in the water, and then after you kind of mix it into the water, go ahead and start smearing your bacteria over your slide. Now, so two important things. One is you don't want to put too much water on your slide. Number two, it's very super important that you guys don't transfer too much bacteria. You want to basically touch the colony, okay? So barely touch the colony that's growing onto your plate, and then go ahead and mix it in the water and spread it all over your slide. Do not pick up the whole entire colony, because if you pick up the whole entire colony, that's going to be way too many bacterial cells that you're going to be transferring over onto your slide, and you don't want that. Remember how I said your smear needs to be thin, And so you don't want to pick up too many bacterial cells, okay? Just by touching it, you are already picking up a whole lot, okay? So as I said, very small amount of water and a very, small amount of bacteria that you want to transfer onto your slide, okay? So just touch the colony and that's it. After that, okay, you want to completely air dry your slides. Now, whether you've already gotten and you did your smear and you got your bacteria from a broth or from a slant or a plate, I mean, at the end, those two slides, they're going to be wet, right? Either because it has water on it or because it has broth on it. So your slide is wet, so you have to completely air dry it. Make sure that you completely air dry it, and you gotta be patient, okay? So don't blow on your slide, don't fan it, because that can aerosolize the bacteria in the air, which is not good. So make sure that you completely air dry it. And the reason why you want to completely air dry it, because we're going to do further steps where we're going to have to rinse our slides several times, and so that can wash. If your slide is not completely air dried, then what can happen is your slide or the bacteria on your slide can actually get washed off and you've lost your sample, okay? After we completely air dry it, we usually heat fix. And I say usually because there are some procedures where we do not do that step, okay, of the heat fixing. So if you do the heat fixing step, again, it's important that you've air dried it first, because if you have some water droplets and you heat fix, that water is going to actually kind of like evaporate, but as it's evaporating, it can actually kind of like, distort your bacteria. So again, it's important that you completely air dry before you heat fix. What is heat fixing? You basically grab your slide and you go ahead and you pass it through the flame 2 to 3 times very quickly. You don't want to hold that slide on top of the flame and just kind of keep it there. That is absolutely, going to be devastating for your bacteria cells. So when you heat fix, you want to heat fix pretty quickly just by passing it 2 to 3 times over the flame. Okay. What's heat fixing for? Well, heat fixing is going to do two things. It's going to ensure that the bacteria sticks to your slides so that you can, that's good because you can then save your slide for later if you want to view it later. That ensures, again, that the bacteria is on your slide, so it's not going to go anywhere. So it's good for that. And then another thing actually is when you heat fix, that kills the bacteria. Okay, so it's kind of, you feel safe then when you're handling that slide because you know that this bacteria is dead. So as I said, so heat fixing kills your bacteria and then permanently adheres it to your glass slide. So we're going to talk about simple stain, and simple staining, this is when we use one dye. And simple staining is great when you want to know your bacteria morphology. Meaning, what is morphology? Meaning shape. Is my bacteria coccus in shape? Is it bacillus? Is it spirillum? Is it vibrio? And then also great for knowing the arrangement and the size of your bacteria. Arrangement meaning, is it arranged in a chain-like format? So that would be strepto. Is it clumped up? So that would be staphylo. Remember those arrangements that we kind of discussed in lecture. So simple stain is going to be able to tell you and give you information about, as I said, the bacteria morphology, the arrangement, its size. Now there are two types of simple stains. There is a direct, or sometimes we refer to it actually as positive. So you might have seen the term positive stain, and we have negative stain. As I said, positive stain is direct stain. So sometimes they refer to it as positive stain or direct stain. With a positive stain or a direct stain, we do heat fix our slide. Okay, so after you let it air dry, we usually go ahead and pass it over the flame 2 to 3 times. The dye that we use with a direct stain or a positive stain is a basic dye. What's known about basic dyes is that they are positively charged. Those are some examples of some of the basic dyes that we usually use in the microbiology lab. Now, a positive dye, what it's going to do is it's going to go ahead and stain the bacteria cell itself, because bacteria cells are usually negatively charged. And so what's going to happen is opposites attract. And so the negatively charged bacteria is going to accept that positive dye, and that's going to stain, therefore it's going to stain the bacteria cell itself. There are lots of basic stains out there. Okay, so there are multiple dyes that we can use that can stain the bacteria. And depending, by the way, on the basic dye that you use, that's going to, again, determine the color that you're going to see under the microscope, right? So if you use methylene blue, well, your bacteria is going to look blue. If you use crystal violet, well, your bacteria is going to look purplish. Safranin is red in color, so if you use safranin, your bacteria is going to look red in color. So depending on the basic dye that you use, that's going to determine the color that you see under the microscope. And as I said, when you heat fix, you're killing the bacteria and you're permanently adhering it to your slide. So therefore, you can actually save your slides if you want to view it at a later time. So saving your slides is great because then you can view it multiple times. You can view it at later times. So that's definitely an advantage. A disadvantage is heat fixing does distort the shape and the size of your bacteria a little bit, okay? So it does distort it a bit. So that's one disadvantage right there of heat fixing. Now the negative stain, it's also a simple stain, but with a negative stain we do not heat fix. We use an acidic dye, and acidic dyes are negatively charged. So as I said, bacteria are negative, and so when you use an acidic dye, which is also negative, what's going to happen is the bacteria is going to repel the dye. And so what's going to end up getting stained is the background, and the bacteria itself, it's not going to get stained. So with a negative stain, you're staining the background and not the bacteria itself. Since you're not heat fixing, the morphology and the size of the bacteria do not get distorted at all. So you can actually view the true morphology and size of your bacteria since they're not getting distorted at all. Well, a disadvantage is because you didn't heat fix. You can't really store your slide because eventually like the bacteria can come off of the slide, and so you can't really store it and view it several times. So usually we view it just once and that's it. You would have to discard your slide. Now, with a direct stain, or as I said, a positive stain, this is just gonna, going over the steps with you. So as I said, we usually use a single dye, and that dye is a basic dye, and we go ahead and we apply it to your prepared smear. So of course, as I said, you grab your slide, you make sure that you've initialed your slide. This slide has to be clean, as I said. Sterilize your loop. Go ahead and put either, as I said, a drop of water and then your bacteria, or you can put a drop of that broth containing the bacteria already. Make sure that your smear is thin. Make sure that your smear is evened out. Then you have to leave your slide to air dry, heat fix it, okay? After you heat fix, you go ahead and you apply your basic dye. We usually leave it for like a minute or so, and then you go ahead and you rinse off, okay, the excess. You rinse off that dye with some water. We usually use distilled water. And then we blot to dry our slides. Okay, so before you view it under the microscope, we usually blot to dry it. We use a special type of blotting paper called Biblios paper. Okay, so Biblios paper, um, that's the blotting paper that we use, and you blot dry it very gently, okay, because you don't want to still, remove your smear, so you blot dry it very gently, and then you're ready to go ahead and view it under the microscope. Make sure that you guys know the steps of how to do a positive stain. Um, Make sure you understand why we're doing each step, okay? Make sure to understand why a basic dye is going to go ahead and stain the bacteria cell itself and not the background. Don't worry about memorizing, like, okay, did I leave that dye for a minute or less than a minute? I'm not going to ask you about the time, but you will need to know the steps, okay, and why we're doing every single step. And then right here, this is an image, you guys, of what you're going to have to do for the virtual labs. You have the Bunsen burner. Here is the, you're going to be picking up the sample from a broth. So as I said, when it's growing in a broth, make sure that before you pick up the sample in here, you either flicker that tube or kind of shake it just to make sure that you are going to be able to pick up that bacteria, because sometimes they settle down at the bottom. And then right here, you're going to be using methylene blue. That's the basic dye, so it's going to be staining the bacteria cell itself, not the background. And this is usually like the bottle of water that we use. We usually work right here on top of a staining tray where we lay down our slide right here, and you kind of do all of your work so that when you go ahead and kind of like you put the stain and you wash it and everything, all of that excess stain and water goes directly into that tray right there. And then here is your loop that you will have to sterilize, dry to Sharpie so that you can label your slides. So again, you're going to be going through that virtual lab to help you out. Another type of simple stain that we're going to discuss is a negative stain. And so same steps though, you have to grab a slide, wash it with water and soap, dry it, okay, after drying it, put down your initials, put my initials right here, put the date. Now if you know what type of bacteria you're looking at, go ahead and write down its name, and then make sure that you also put down like, okay, what type of stain are you performing, since this is a negative stain. Now with the negative stain, the procedure is a bit different. So instead of putting a drop of water on our slide, we don't do that. We put a drop of the dye right away. So you put a drop of the acidic dye. Remember with negative stain, we use acidic dyes. So an example of an acidic dye that we usually use in lab, it's called the nigrosin dye. There's another one called India ink. So again, there are a bunch of negative stains out there. So nigrosin dye, it's black in color, so we usually put a drop of it, and we usually put it on the side, not in the middle of the slide, like right here on the edge, okay, of your slide. So on the edge of your slide, you put a drop of the nigrosin dye, and then you grab your sterilized, and here's another image for the procedure. You grab your inoculating glue. You go ahead. Let's say you're grabbing it from a slant, or from a plate, for example, or from a broth. Go ahead and just put an inoculum of your bacteria into the nigrosin, and then go ahead and kind of mix it in. Okay, so mix it in. But then this time, don't use your inoculating glue to smear it all over your slide. we have a different technique where you grab another clean slide. Okay, guys, like right here, grab another clean slide, and then you're going to take that slide. You're actually going to smear the dye with your bacteria down the length of your slide, kind of like this. So let's say here, again, here I have my initials and everything, and then you put like your nigrosin with the bacteria right here. You go ahead and you kind of, smear it down the length of your slide. So it's gonna look something like this, okay? Of course, as I said, you never smear it where you have your initials. So on the other side, you're going to go ahead and smear it down the slide, and that's it. That would be it. And then you will have to leave your slide to completely air dry. Okay. After it completely air dries, then you are ready to view it under the microscope. And then over here, there are a list of some videos for you to watch on top, of course, of the virtual lab that you will need to do for this week. Over here, you have two images, okay? You're going to have to figure out which one is the negative stain, which one is the direct or positive stain. And then you have some questions to answer about those two images for week 5 worksheet.